substitution, how soils have developed over 6,000 years, and how the vegetation responds. And so we've got increasing soil age. We can start to understand how soils influence plant communities. And just very quickly to show you how the soils look over a few thousand years of soil development in this ecosystem. On the left here is a 300-year-old soil. It's formed in coarse sand. You can see an A horizon's formed and a very slight weathered B horizon starting to form there. But after a few thousand years, those processes have created this soil. This is a podzol, if you know your soils. This is a bleached E horizon where this organic horizon has leached out the iron from the soil, leaving a white, sandy horizon, and it's formed here a pan, a hard iron pan, and roots can no longer get through here. And so we've got changes to the hydrology and therefore the ecology of these forests. So that's what 4,000 years of soil development looks like in that environment. We can also find chronosequences in other places. For example, there are some very interesting glacial sequences. Glaciers are retreating all over the world. And so as they retreat, they leave behind moraines, datable moraines. And we can go back down, for example, in this case, down the valley, finding moraines of different ages. In this case, you can't probably see that, but up to about 100,000 years old. And so we can go to those moraines, look at the soils, and link this to weathering. And you can see the total phosphorus declining, the total nitrogen accumulating, and the entropy ratio, the relative availability of nitrogen and phosphorus to plants increasing. And you can't see that because it's right behind me. Now, there are relatively few long-term chronosequences around the world, but I put some up here on different soil parent materials, different climates. There are glacial sequences. There's a good one in China, just close to Chengdu on the edge of the plateau. There's the classic Hawaiian Islands sequence of volcanic soils that Peter Vertusek and colleagues did so much work on. Sand dunes in Australia, terraces, marine terraces, Swedish islands, all these places where we find long-term sequences of soils that vary only in time, with all of the soil factors held constant. And so wherever we find these chronosequences, we find very similar and consistent patterns of soil development and of phosphorus and nitrogen availability. Now, I started off this little section talking about diversity. So the question is, how do these patterns influence plant diversity? And I want to take you to this chronosequence to address this question. Of all those sequences, this one probably looks the least interesting. It's muted topography, low diversity, shrubland, doesn't look very interesting at all. But actually, this is one of the best places in the world to study how soils are influencing the diversity of plant communities. It's down there on the west coast of Australia, just north of the city of Perth. And it's interesting from a plant perspective because it's a hotspot of diversity, and a global hotspot of diversity, not just for species richness, but also there occur nowhere else. And so the plants are interesting, but also the soils are interesting as well, because on this coastline, we find some very consistent 2 million year sequences of soils. Just to explain very briefly how they're forming, they're sand dunes, which form during interglacial glacial cycles. So if you look on the image at the top here, this is the Delta 18O of benthic foraminifera. This is showing this glacial interglacial cycle. The blue, the positive Delta 18O is a glacial period when there's lots of ice on the land in the northern hemisphere. Sea levels go down. Then as we come into an interglacial, sea levels come back up, and all that material that's washed off the Australian continent is then moved back against the coast and forms a dune. And so we get this series of dunes, each one related to a glacial, interglacial cycle, going back to the start of the Quaternary about 2 million years ago. So we can go, just like we did at the New Zealand sequence, to each of these dunes, look at the soils and look at the plant community and understand something about how the soil changes are affecting the plants. Why my presentation doesn't like to move on. Hang on. think it just needs its thinking. I don't know why. My apologies. Ah, well done. No, that's good. Oh, I lost it. Okay. Yeah. Okay. Just to show you very briefly how the soils change over 2 million years of pedogenesis. On the left, you can see young soils. These are Holocene soils formed since the last glacial period. They're basically formed in carbonate sand, so they're full of shell fragments and other material that comes from ocean organisms, and so very high pH, relatively high phosphorus. We get these white, deep profiles, basically of quartz sand. You can see the start of soil development there with an A horizon forming at the top. As soils develop, they acidify, and that dissolves the carbonate, and so the carbonate is lost, is leached from the profile, and we're left with soils that look like this. The orange color is iron oxides, which coat sand grains. And so basically we've lost the carbonate and we're left with these residual orange sands. And we've already lost most of the phosphorus at this point. And these are middle Pleistocene soils, about 100 to 500,000 years old. And then over time, that process of podzolization, the organic matter, pulls those iron oxides off the sand grains, off these quartz grains, and leaves us with something like this. It's very similar to the soils that we heard about in Madagascar earlier, these deep bleached quartz sands. Basically, these are some of the lowest phosphorus soils in the world, and I imagine that yours are similar in Madagascar. So we go over 2 million years, we end up with these incredibly low phosphorus, incredibly infertile soils. And what about the diversity? Well, the patterns of diversity are very clear as we go through this sequence. As phosphorus declines over 2 million years, the diversity of plants in this ecosystem increases. And I plotted here 3 different measures of diversity, the alpha diversity in the red circles there, increasing from, this is the plot level diversity, and all the work that I'm showing you is based on 10 by 10 meter plots in these shrublands. So the plot alpha diversity increasing from about 15 species up to around 50 species across this gradient. The gamma diversity, so when we add up the diversity in those plots, the species in those plots for each dune, the gamma diversity increasing from about 30 to more than 150 species. And the beta diversity increasing similarly very strongly across the gradient, both for all species and for woody species. So we find this very clear pattern of increasing plant diversity with increasing soil age. So as phosphorus is declining and becoming very limiting on these old soils, the plant diversity increasing. And you may think this is related just to this particular system, but in fact, when we go and find and look at plant diversity on chronosequences in other areas of the world, we find corresponding patterns of increasing diversity, increasing alpha diversity. It doesn't matter if we're on the Swedish islands, Australian dunes, glacial chronosequences, the Hawaiian islands. In all these cases, diversity is increasing through time. Low phosphorus is associated with high plant diversity. And so going back to my original slide about the latitudinal gradient of diversity, if you think about where the high diversity is in the world, in the Amazon basin, Central Africa, Southeast Asia, these are all places in red here where we have the oldest, strongly weathered, lowest phosphorus soils. And so perhaps soils somehow, I don't have time today to talk about this mechanistically. We thought about this a lot, but low phosphorus is absolutely associated with high diversity. So the second question is, how do soils influence the distribution of tree species in tropical forests? And this and the next part I'll talk about is something I did in collaboration with Rick. Actually, Rick probably did most of the work, so I should acknowledge his contribution to this before I go on. To talk about this, I want to take you to Panama. Now, this is Panama. If you're not aware, it's down here, bridging South and North America. It's a really interesting place for studying plants. Of course, the first large plot was there on BCI, but it's really interesting in general from a soils perspective, just because it's very diverse topographically and very diverse geologically. And so I'm going to zoom into this. Sorry, there we go. I'm going to zoom into this area here. This is the Panama Canal. We've got Panama City here. The ships come through the canal here into this lake and move out into the Caribbean. There's Barra Colorado Island, which I showed you earlier. And what you can see here is a legacy of the canal, of the Panama Canal, which is a 10-kilometer wide strip of forest, of protected forest, running across from the Pacific side to the Caribbean side of Panama. The interesting thing is that this also spans a very strong rainfall gradient, from less than 2 meters per year on the Pacific coast to more than 4 meters a year on the Caribbean coast. And it's also a very diverse area. So I mentioned earlier, the BCI plot has 300 species, but there are many, many more species in this region, in this protected forest. And from a soils perspective, this is also fascinating because the geology is very complicated in this area. So without mentioning what's what here, I hope it's obvious that there's a very complex geological situation in central Panama there. We've got volcanic material, sedimentary, limestone, all kinds of soil parent material, and therefore a real range of soils. And there's an example of some of the soils in this area. Many of these deep, red, strongly weathered soils, but also a real variety. We have high phosphorus to low phosphorus, high pH to low pH, a real mixture of soil properties in this region. And so how do we start to understand species distributions in this region? Well, the first thing was moisture, and it was known for a fairly long time that moisture was really driving the distribution of many of the species. And this is an example of one species. This is Xylopia macrantha. It's a relatively drought-sensitive species that occurs widely in Panama. And what I'm showing here on this top graph is the BCI plot here. And the circles are small 1 hectare plots that Rick established many years ago across this rainfall gradient. All the filled circles are a plot where this species occurs. So all these places contain a Xylopia macrantha, and the open circles contain this species doesn't occur. So what you can see very clearly is that this species occurs on the wetter Caribbean slope and not on the dry Pacific slope. And this is the 50-hectare plot. Each black dot is an individual of this species, and it occurs predominantly on these southeastern slopes where moisture tends to be a bit higher in the dry season. So an example of a drought-sensitive species, and it was possible to do this analysis for many hundreds of species that occur in these plots. So we had a fairly good understanding of how moisture influenced species distributions. But then we could do the same thing for nutrients, for soil nutrients, and in this case, phosphorus. And I'm showing 3 examples here of species showing contrasting responses to soil phosphorus availability. And so the first, what I'm showing here this is resin phosphorus. This is a measure of readily exchangeable phosphorus in the soil. And on the y-axis is the probability that a species occurs in a plot. And so the first species is Cavanalesia platanifolia. This is a canopy emergent, deciduous canopy emergent, occurs widely in Panama. And what you can see is at low phosphorus, low concentrations of phosphorus, this species never occurs. And then above about 2 parts per million of resin phosphorus, we find Cavanalesia occurring. And in fact, on the highest phosphorus soils, you're almost guaranteed to find Cavanalesia platanifolia. And so we take the first order coefficient of this model and call this the effect size, or the phosphorus affinity, in this case +2 for Cavanalesia. That's a strong phosphorus favoring species. Now the opposite pattern is this one for this Eschwellera species. You can see that at high phosphorus this species never occurs, but when we get below 2 ppm we have a very good probability of finding this species, and therefore it has a negative phosphorus association. It does not like high phosphorus, and so 2 contrasting affinities for phosphorus. And in the bottom, this is a species that doesn't care about phosphorus. This is Ingavera. It's a nitrogen-fixing species, and you can see that it occurs across the phosphorus gradient. It doesn't care about phosphorus. It actually has a strong moisture affinity. And so 3 different contrasting responses to phosphorus. Come on. I'm very sorry about my talk. I don't know why nobody else has had this problem. I don't know what's happening. Just seems to. It's thinking, but it doesn't like it. Okay, as we get back to that slide, the upshot of this is that we can start to understand the distributions of the hundreds of species in this region based on 2 key variables, on the moisture and the phosphorus. Perfect, exactly. And so what we've plotted here is the moisture effect size, this whether the species prefer high or low moisture or don't care about moisture, and the phosphorus effect size, the high phosphorus loving species, there's Cavernalesia +2. The low phosphorus species, there's Eschwellera at -2. So we can start to understand the distribution of the hundreds of species in this region based on just on these two variables. And it turns out that about 2/3 of the species have a strong moisture affinity, and about 60% have a strong phosphorus affinity. So we really start to. This is one example of how we can start to use soils to start to understand something about the distributions of species in these very complex communities. Oh, it's coming. Just to show you very briefly, there's an example of Cavernalesia platanifolia. It's Bombycaceae. Rick's probably immediately saying this is not Bombycaceae anymore. I think it changed to Malvaceae. Okay. Yeah, so we don't. Anyway, there it is, a beautiful canopy emergent tree. And on this side, this is an example of how strongly this species is faithful to its phosphorus association. So this is an example of the banks of the Panama Canal. On this side, no Cavernalesia. On this side, you can see abundant Cavernalesia in the dry season. This is a fault. On this side, we have basaltic soils, which are very low phosphorus, no Cavernalesia. This side is marine sedimentary soils, high phosphorus and covered in Cavernalesia. So just an example of how strongly these species stick to their associations. And so come on. Okay. So the question is why? Why do these species have these associations? This is a question I can't answer, but I can give you an example of a possibility. One of the things we find, one of the interesting things is that there are many congeneric pairs of species showing very different affinities, different associations to environmental variables. So for example, many congeneric pairs of species have opposite phosphorus associations. One will be high phosphorus, one will be low phosphorus. And this is a good example of 2 species of Tababuia, Guayacan and Rosea, a low phosphorus specialist and a high phosphorus specialist. And of course, these trees are planted widely in botanic gardens and things. You may have seen them around here. Now, why are these? What's different about these species in terms of phosphorus that might allow this to happen? And one of the possibilities is that Tababuia guayacan is something they call a pollen cheater. A pollen cheater produces very low amounts of pollen. And of course, pollen contains very high concentrations of phosphorus. And so one possibility is that this species is saving some phosphorus or helping to be more phosphorus efficient by making less pollen. It relies on the fact that the insects can't remember last year that they didn't get enough pollen, so they come back. The tree gets pollinated, but its investment in pollen and therefore its investment in phosphorus is relatively low. So that's just one possibility of how these species might somehow save phosphorus. There are many others, but this is something to be studied. My final and third question is, does phosphorus limit tree growth in lowland tropical forests? This is about productivity of tropical forests, and it's been considered for many, many years based on this idea that tropical soils are all old, strongly weathered. Phosphorus must be constraining growth. These plants must be limited by phosphorus. And there is indeed lots of evidence for this. We do find strongly weathered soils. We do find low phosphorus concentrations. Trees have wide nitrogen to phosphorus ratios in leaf tissue, suggesting phosphorus limitation. But we also, there are questions about nitrogen. Nitrogen is assumed to be not limiting, but we find we have this paradox of legume abundance. There are many species and many stems, particularly in the neotropics, in the legumes, so that trees which have the capacity to fix nitrogen. And so if nitrogen is not limiting, the question is why there are so many legumes in these forests. But surprisingly, the experimental evidence for this phosphorus limitation is really not there. And in fact, there have been many studies now where people have tried to experimentally add and have experimentally added nutrients, nitrogen, phosphorus, potassium, to forest plots, to tropical forest plots, and tried to measure a change in growth. And in fact, there's only one experiment that found a change in growth with phosphorus addition, and that was on the Hawaiian Islands, on the old soils of the Hawaiian Islands chronosequence, where they found a response to phosphorus. But this is an unusual place because, of course, Hawaii is very low at diversity. It's basically dominated by a single tree species, Metrosideros polymorpha. And in mainland locations, in diverse lowland tropical forests, there's been no response to phosphorus. And I know this very well. I worked with Joe Wright for many years on a long-term 20-year experiment in Panama that involved nitrogen and phosphorus and potassium additions. And that was basically the biggest null result in my career, because there was no response to any of those nutrients in terms of trunk growth. So it seems that perhaps this idea that phosphorus is limiting tropical forest growth was not correct. But of course, we have this wonderful phosphorus gradient in Panama across this strong rainfall gradient, where we can approach this question of phosphorus limitation in a slightly different way. I mentioned Tababuya Guayacan, so I'm sure you're all thinking immediately, this is a very low phosphorus part of Panama, the Tababuya Guayacans everywhere, the yellow flowers. We can go to those plots, measure growth repeatedly to quantify growth rates and try and understand whether trees and growth rates of individual species are responding to phosphorus. And very simply, this is the result. What I'm showing here is growth responses in millimeters per year to phosphorus for large trees and small trees, and responses to moisture, again, for large trees and small trees. And the effects of phosphorus, this is phosphorus in the plots, in those 1 hectare plots that I talked about earlier. Basically, the response is small, but it's very consistent. And you can see the faint gray lines are individual species. You can see most species responding to phosphorus in their natural ranges by increasing their growth. And the black lines are the meta growth response, the average of all those species. And you can see for both large trees and small trees, we have a very clear and consistent increase in growth under natural conditions to phosphorus. Moisture has no effect on growth for the large trees, but an effect for the small trees, which we always interpreted as the fact that small trees have much more shallow root systems and are therefore more sensitive to the dry season. And actually, this occurs really across species. It's a fairly pervasive response, because we see, if we look at responses of small trees and large trees to phosphorus, only 3 species out of this couple of hundred common species don't respond to phosphorus either as young trees or as mature trees. And so almost all species growing faster with more phosphorus in their natural ranges. And there's a couple of other interesting points that came out of this analysis. The first one is this. This is the phosphorus affinities, the phosphorus associations that I explained earlier, from the low phosphorus species to the high phosphorus species against their observed growth rates. The blue circles are species means, and the black dots there are individual stems. And so surprisingly, we find that the fastest growing species on average in that central Panama region are the species which favor the lowest phosphorus soils. So you might expect that as species are growing faster with more phosphorus in their ranges, that we would find the highest growth rates for the highest phosphorus species. But in fact, it's the opposite. And so this is somewhat paradoxical. We find that the fastest growing species on average, are the ones that are on the lowest phosphorus soils that specialize at low phosphorus. And finally, when we add up the growth in those plots of all the species and calculate a community-wide plot level growth, it turns out that there's no variation across that gradient. So somehow the responses of individual species growing faster with more phosphorus, this does not translate into a community-wide growth. So basically growth rates, plot level, community-wide growth rates consistent across that very strong phosphorus gradient. This is a real challenge to modelers who are trying to understand how growth of forest will change in the future in response to global change drivers. That's everything. I hope I didn't take too long with all those problems with the slides, but just to summarize very quickly, I talked about pedogenesis and plant diversity, and I showed you that the highest diversity, when controlling for other factors, for climate, for relief, for topography, the highest diversity plant communities always occur on the oldest soils, and that applies in ecosystems worldwide. I showed you that species distributions in very complex and very diverse tropical forests can be understood primarily through two measures, through moisture and soil phosphorus availability, and finally that productivity, that phosphorus does limit growth in tropical forests, but at the species level. And so this does not occur and does not translate into a consistent community-wide growth. Okay, that's me finished. Thank you very much.
Okay, thank you very much. We have time for maybe one question, if someone has a question. Yes.
Hi, Benjamin. Thank you very much for your very interesting talk. I noticed that you said moisture and phosphorus determine the tropical tree species distributions, and you also showed some slides about the relationship between phosphorus and the moisture with the growth. So how do you monitoring the moisture? How do you define the moisture here? You measure the soil moisture continuously, or you did some treatment? And how about the phosphorus? You add phosphorus, or you just investigated the soil phosphorus?
Yeah. So so there's a very strong approximate 3-month dry season in that region, but it's a much stronger dry season at the lower rainfall site and a weaker dry season at the wetter sites. And so we modeled the moisture availability based on a network of weather stations. So the Panama Canal has a very dense network of weather stations because they're monitoring water inputs to the canal. And so we can use that data to calculate very precise estimates of rainfall and soil moisture at each site. So the moisture is modeled, the soils are measured. So we go and collect soil samples from across each one of those 1 hectare plots and quantify nutrients. I didn't talk about other nutrients, but we did nitrogen and cations and many measures of phosphorus. And the measure that always comes out as the most sensitive is that exchangeable phosphorus, that resin extractable phosphorus. Yeah, it explains many, not just this, but many variables, many ecological properties that we find in that region. So yeah.
Actually got
Yeah.
Yeah, we did some treatment.
Mm-hmm. Okay. Sounds interesting. Yeah. Okay. Great. Yeah.
Okay. We have to close the questions here and move on to our next speaker. But Ben, thank you very much. And our next speaker, Dr. Keeping Ma, one of our board members. He's actually been with the journal since its inception, and certainly not a stranger to many of you here today. Very well known for his botanical work in China and around the world. So with that, Dr. Ma.
Thank you very much. it's really an honor for me to present a talk to introduce the network related to forest biodiversity monitoring. Maybe I can say to Rick, this is a kind of report how China learned from CTFS. And then we also have some development. This is a slide to show the development. And as for biodiversity monitoring, and we in 2015 we set up so-called Sinobond, and including forest biodiversity monitoring, we call it C4Bio. And besides C4Bio, we have other thematic networks such as mammal network, bird monitoring, amphibian reptile monitoring, fish monitoring, insect monitoring, soil invertebrate monitoring, and also for steppe and desert monitoring, canopy biodiversity monitoring. And we also have soil microbiome monitoring. So this is totally 10 thematic networks for biodiversity monitoring at the national scale of China. And we also have synthesis center and try to develop a standard and criteria for monitoring, and also for management and sharing, and also for remote sensing, in particular near surface remote sensing. So this is the general structure of the biodiversity monitoring in China, developed and maintained by Chinese Academy of Sciences. We also publish a pretty number of papers to introduce the network, the plan, the achievements. But at the moment, most of them are in Chinese. And as for C4Bio, we have this map to show the structure or distribution of the forest dynamic plots in China. Totally, at the moment, we have 30 forest dynamic plots. And we also have 61 to 5 hectare plots. 30 forest dynamic plots refers to large permanent plots. Here, large refers to the size of a plot is around 20 hectares. And from that slide, you can see the total area of the plots is about 800 hectares. The total number of woody species tagged and mapped with DBH more than 1 centimeter is 3.1 million, which belongs to 3,000 woody species in China. This is a general description. And we also try to collaborate with neighboring countries and to extend our network into the typical tropical forest areas. And so from that side, you can see we collaborate with colleagues from Thailand and also Malaysia to set up a really local network in tropical areas. Oh, you're 20. And I think these slides you might have seen for many times. That's why mapping biodiversity, and to explore the key question, how do all the different species coexist in the same forest? That is the critical question for setting up forest dynamic plots to explore. And I just take Qianjiangyuan National Park pilot area as an example to show what we are doing with the forest dynamic plots in China. And for monitoring of forest, first we try to use near surface remote sensing approach to scan the forest around the permanent plots. Based on the data, we can produce such kind of maps for DEM, for leaf area index in the protected area. The size of the protected area is 252 square kilometers. And we also have that kind of map for canopy gap fraction and also for canopy cover. And also for mean tree height standard deviation, the variance of the tree height in the site. And we also set up different size of forest dynamics plots in the protected areas. And totally, we have set up more than 700 plots in the protected areas. And 252 square kilometers. That means per square kilometer, we have around 3 such kind of plots on average. And for the plots, we have 124 forest dynamic plots, 5 hectares plots, 13 1 hectares plots, and 27 30 by 30 permanent plots. And we also have, say, more than 700 20 by 20 hectares square meters plot. And based on this data, we can produce such kind of maps, such as evergreen tree dominant species at the canopy layer, and also the Chinese endemic plants distribution in this area, and also the threatened plants in this area, to produce such kind of map, and also to show the distribution of threatened plants in this area. And based on both the remote sensing data and also the plots data, we produce a vegetation map of this area, which is very important not only for research, but also for the conservation planning and management of the protected areas. And this is the site to set up the 24 hectares permanent plot or forest dynamic plot. That is 400 meter by 600 meter. That is 24 hectares. And this is the topography of the plot. And by using total station to make the grading of the plot by 20 by 20 square meters. And after the first survey of the plot, we can produce such kind of map, and say for each unit plot, we can have tree species richness. And then for 24 hectares, we have such kind of map, and also for abundance of the trees in the plot. And we also set up seed traps and seedling plots. This is the stations for, we call it the Gutianshan Forest Dynamic Plots. The total number is 169. And for each station, we have 1 seed trap and 3 1 square meter seedling plot. Here is the seedling plots. We marked every seedlings in the 1 square meter plot. And also this is the seed trap. It is the same size, same shape with those produced by CTFS. And we also set up 3,000 dendrometers to monitor the annual dynamics of DBH of 3,000 trees in this area. And so from these slides, you can have the view that we not only monitor the trees, but also seeds, seedlings, saplings, and adult trees. So that is try to monitor the whole forest. And then we also have soil mapping. It's about 23 property features of the soil. And we set up, we collected around 1,000 samples from the 24 hectares plot. And this is the 13 1 hectare plus distribution in the protected area. And for we also conduct wildlife camera trapping, and that's also very interesting and part of the ecosystem. And first, in the 2014, in May of 2014, we set up the first group of camera traps. It's around 100 sets. And for each 1 square kilometer, we set up 1 set of camera trap. And from the collection of data, we can have this kind of map to show the distribution of different species in the protected area. And also for different species, for different year, you have this kind of map of the distribution of a particular wildlife. And this is the national, say, first category of national protected animal, that's black muntjac. And you can see this species distributed only in the core area of the protected areas. But the species, another species of the same genus, not only distributed in the core area, but other areas even outside of the nature reserve. So this kind of data is really welcomed by the local manager of the protect area. And also to produce such kind of map by using the vegetation map and also the distribution data to produce such kind of map to show the suitable habitat for this species. And this is the slides to show the dynamics of population of the wildlife in the nature reserve. I think it is also very interesting. You can see most of the species population is increased. That's to show the effect of management of the nature reserve. And we can also calculate wildlife picture index. This is to show the dynamics of the community, the diversity, and not only the population dynamics of a particular species. This is a community indicator. So from this slide, you can see from 2014 to 2022, the community diversity is more or less stable. So that to indicate the management effect of the nature reserve. And based on the data from this forest biodiversity monitoring network, C4Bio, we have published more than 900 papers, and among them, more than 500 published in international journals. And some of them are published in high-profile journals. And this morning, Professor Chen just introduced a case based on one of the plots, Nabanhe forest dynamic plots in the network. And we also publish more than 10 stat books for each forest dynamic plots. We try to produce one book to introduce the every species mapped and tagged in the plot. So that's also very helpful. And this is the traditional homework for learning CTFS to set up Chinese networks for forest dynamic plots. And we try to develop branch networks. This is one of them, the biodiversity and the ecosystem function manipulation experiment network. And from this slide, you can see so far we have 12 such kind of BF experiment sites. Just take, say, this BF China as an example to show what we are doing with the manipulation plots. And over there, it is the distribution of such kind of plots at the global scale. And in China, we set up the BF China plot. And this plot is very close to the site I just introduced. It's about a 1 hour drive. And we have two sites, site A and B. And for site A, we set up in 2009. For site B, 2010. And for the manipulation experiment plot, we have set up 566 study plots. Each plot with the size 666 square meters. Why such kind of number? That's because the traditional Chinese unit, we call it mu. 1 mu equals to 666 square meters. And for site A, that is 18.4 hectares. The elevation is 105 to 275 meters. For site B, that is 20 hectares. So we use more than 38 hectares, but we rent 50 hectares for the experiment. And we have selected 42 tree species, 18 shrub species as species pool for set up the experiment. Totally, we have planted 220,000 trees, 88,000 shrubs, totally around 300,000 woody species in 2 years. It's a really challenging job, and we organize more than 100 local farmers to help us to plant trees in 2 years, 2009, 2010. This is the idea to design the experiment. And we rent 2 nurseries to produce seedlings. And because only 2 species you can buy seedlings from the market. Most of them we should collect seeds ourselves and then to produce the seedlings by the nurseries. And we also try to grade the size, and also try to write down the names of each of the species names, and then on the bamboo shoot, and then to place the bamboo shoot in the right point, because we have different levels of diversity. 1 species, 2 species, 4 species, 8 species, 16 species, and 24 species. This is the most complicated design of its kind at the global scale. From this slide, you can see we really hire many people to help us to plant trees. As you can see, it is really challenging, not as a regular planting trees for forestry department. And we have different levels of diversity. For each point, we have a fixed species of seedlings. So that's really a difficult task. And we are very happy to see the trees really grow very well. So from this picture, you can see some of the trees, in particular deciduous trees, grow very fast. And some of them are nearly 30 meters high. And if you have interest to know about the design of the site, and you can learn something from the paper published in ME. And I also published a paper in Chinese to introduce the experiment. So our Chinese colleagues can download this from the Journal of Biodiversity Science. And I would like to introduce some of the progress of the research. And so far, based on this experiment, we have published 260 papers, and among them 240 in SI index journals, and including Science and Science, Nature, family journals, about 20 papers published in such kind of high-profile journals. And I just briefly introduced 4 different kinds of progress. And first, biodiversity and ecosystem functioning along the standard age. And this is a paper published in Science in 2018. And from the slides, you can see the biodiversity and ecosystem function here refers to primary productivity. These two are positively correlated. And we use huge amount of data to confirm this. And also from these slides, you can see, the positive biodiversity effects increasing over time. That's also important for this research. And for BF research, there are really, huge discussion on which parties contribute more, complementarity effect or selection effect. And from our experiment, we confirmed that the biological complementarity effect is increasing over time, and the selection effect decrease over time. This is also an important conclusion. And also for function diversity and productivity, this is the first time to show the temporal dynamics between forest functional characteristics and the productivity has really positive correlated. This is the first time to show this. And I just like to briefly introduce. And we select 38 functional traits and to carry out analysis. And from these slides, you can see we set up 2 different indices, effect size and reliability, and both to show the functional traits diversity and primary productivity are positively related. And not only use one group of data, but we use 10 replicates to calculate the data, and then we have confirm the conclusion. And we also try to use this experiment data to analyze the mechanism of the stability of community. And this is the results. Stability, the ability of forest to maintain functioning over time, in particular in the face of environmental stressors. The main mechanism is that for different species in the community might respond to the environment change differently. And in total, if you consider the community, different species of a community together, and the community level, it might be stable rather than a lot of variation for a particular species in the community. And this is the slides to show the community stability significantly increase with species richness. And also for the overall annual survival rate was marginally higher in more diverse than less diverse stats. So we have such kind of experiments with different levels of diversity. I just said 1 species, 2, 4, 8, 16. So we can have the data to carry out such kind of analysis. And for the second topic, tree genetic diversity and productivity, this is also a special feature for the experiment. We collect data for different trees. And seeds from one tree, we call it one seed family. And you can regard it one seed family as a species. And we have an experiment with different level of species. And to do the same, we can have different level of seed families and to show the effects of genetic diversity. And very few studies on this kind of topic. And from these slides, you can see genetic diversity promoted the variation of functional traits within individuals and then result in varied resource utilization and ultimately significantly affected community productivity. And so this is to say genetic diversity and productivity are positively related. And not only the diversity, but also genetic identity are related to productivity. And the paper last year just published in New Phytologist. And the third topic is about multi-trophic diversity and productivity. That's also very important. And we talk about plant and plant-plant interactions. That is regarded as horizontal diversity. And in the ecosystem, we also have vertical diversity. That refers to different trophic levels diversity. And the German colleagues also working in the platform published a paper in Nature Communications to compare the two very important BF manipulation experiment. One is the BF China experiment, another is Jena experiment, that is about grassland BF research. And from this, you can see we talk about, say, herbivore, predator, and parasitoids, different levels, different tropical levels diversity, the relationship between these. And just as I mentioned, say, for most of the BF research talk about the relationship between plant species richness and the primary productivity. And just regard the tropical level contribution as a black box. So we try to open the black box to see the contribution of the different tropic levels. How about the contribution? And this is the different ways to collect data for the different tropical levels. And from the observed data, we have these kind of results. Say the multi-trophic species richness has a positive relation with tree species richness, and further multi-trophic species richness have positive relation with primary productivity. And we also try to analyze the, say, relationships among different taxa or trophic level. And we just divide the insects part into two different trophic levels. One is a herbivore, another is the enemy of herbivores. And say from this figure, you can see tree functional diversity and tree species diversity has a positive relationship with herbivore species richness. If you only look at this part, you might have the image that herbivore diversity might reduce primary productivity. And if you try to include the enemy of herbivore, and then you can understand the whole picture. Say, tree functional diversity also has a positive relationship with the enemy of the herbivores. And then the enemy can control the herbivore level. So the whole story is that tree functional diversity and species diversity interact with herbivore level, herbivore enemy level, and then we have, say, positive relationship between tropical diversity and tree diversity. And we also carried out research on multi-tropic diversity and multi-functionality. And first, we also carry out analysis with forest BF experiment and the Yena grassland experiment. And then we carried further analysis and have the result as the association between multi-diversity and multifunctionality is stronger than the relationship between the diversity of single trophic groups and the multifunctionality in both ecosystem types, forest and grassland. And also, plant diversity increases multifunctionality through elevated multi-diversity or interactions among trophic groups in both ecosystem types. And the multi-diversity and the tropical interactions play a more important role in influencing multifunctionality in more complex ecosystems. And this is to show one of the branch manipulation experiment. And also we set up another branch network that is forest crane network. We have set up 8 such kind of forest cranes in the forest dynamic plus site from temperate to tropical. And this is the forest crane in Dinghushan. This is the forest crane in Xichuanbana, because the trees is much higher than in Xichuanbana than in other sites. So the Xichuanbana forest crane is 80 meters high. The arm is 60 meters long. For other sites, the height is 60 meters. And we also set up branch networks for phenology. And at the moment, about 20 forest dynamic plots joined the phenology network, and from cold temperate to tropical forest. For the phenology observation network, we have set up such kind of infrastructure for phenological camera, radio growth meter, and meter rhizotron technique, and the corresponding indicators. We try to collect data to show the dynamics. And we also organize a lot of workshops for the network. This is the workshop for wildlife monitoring with camera traps in 2014, and the second and third workshops. And another series is about application of LiDAR in forest biodiversity monitoring since 2015. And this is the second workshop, also for exercise after giving lectures. And the third workshop on application of LiDAR, and also exercise. And the third, say, group photo. And the fourth, and the fifth. And we also organize a national symposium on biodiversity monitoring since 2015. This is the first one, and this is the second one, and this is the third. This is online, and this is the fifth. Every 2 years, we organize a national level symposium on biodiversity monitoring. And we have much more, but in Chinese, say we have, say, a joint workshop by CTFS and CIFOR-Bio, financially supported by both NSF from China and the United States. And first, we applied five workshops once each year, and approved. After 4 workshops, the 2 foundations agreed to extend by 2 joint workshops. So that's very well received by participants from different countries. And we also have, say, cross-strait workshop, and for 10 such kind of workshops. And we organize a pretty number of training workshops, and also for the CIFOR-Bio forum to invite colleagues to deliver lectures. And say we invited a lot of foreign colleagues to present talks in the forum. And if you have interest to learn more about what happened about biodiversity science in China, I just organized a team to prepare a review paper and publish in National Science Review. That is a good journal. If you value the good or not by using impact factors, and the current impact factor is 16, so that's pretty high. That's a freely download paper. And I just like to add 3 points why, say, Chinese network are attractive to a lot of colleagues. And first, I think, say, when a team for a plot join the platform, they have more opportunity to share data with other forest dynamic plots. And second, this is a community. When you join the community, you have more opportunity to showcase your research, because we have more communications within the network. And the third, I think it is also very important for data sharing, for training, for international collaboration. The network is much more better than an individual or a small team. So I really hope more and more colleagues and teams and institutions join us and collaborate with us. Thank you.
Okay, so in the interest of time, if you have questions for Dr. Ma, you can catch him at the break. We're supposed to be back at 15:20, but let's call it 15:25 so that everyone can get a snack and come back. Okay, thank you.
We started the computer.
Yeah.
对对对，我要重启一下。Restart.
these people start finding you sick. Okay. Oh. Okay, and we are now start the second session for this afternoon, and understand the weather is getting warmer, and so after lunch and also the break, and the brain is running a bit slower, and at least for me, and so, but for the last session for today, we have two rising scientists and with brighter minds and with promising career. And first I have Dr. Xu Han, and from Institute of Tropical Forestry Research, which is an institute affiliated to Chinese Academy of Forestry. And Xu Han obtained his PhD from Chinese Academy of Forestry. Since then, even during his PhD study, he's been working with tropical forests, and his institute based in Guangzhou, but most of his work is in Hainan Island. And so I'm saying young, it's relatively compared with people like me, but he has more than 10 years of research experience with tropical forests, and he's not only in ecology. During his work, he discovered 7 new species, named 7 new species from tropical forests. And now I hand over to Dr. Xu Han.
Thank you very much for the so detailed introduction. And it's my pleasure to be here to share our researches in Hainan Island. I will just share one interest in the animal behavior that in Hainan Island. And thank you very much for the invitation from the Forest Ecosystems Journal and by the Professor Rick and so. And for my presentation, I will introduce. So firstly, I will introduce, give a brief introduction for our research in Hainan Island, and then second, I will turn to our study on the squirrels and the nuts. Because maybe tomorrow we will go to Hainan Island, so I want to give you some brief information. This is the Hainan's location that Professor Liu has introduced this morning. And we know that the Hainan Island, the latitude is valid from the 18 to 20. And the main Hainan Island area is about 34,000 kilometers square. It's the second largest island of China. And Hainan in 1988, it's belonging to the Guangdong province. But after that, it's separated and into a different province. Now we call it the Hainan province. And on the on the north, it's the Guangdong province. The latest Chunzhou Strait between the Hainan and Guangdong province. And for how the Chunzhou Strait and Hainan Island formed, there are several different series. And the common idea is that the rising sea levels flooded and low-lying land between mainland, between Guangdong and Hainan. But the newest finding is that the Hainan Island is rotated 150 degrees and drift to the southeast direction. And for the topography of Hainan Island, they are about 39% of the mountains in Hainan Island. The highest mountain in Hainan is Wuzhishan. It's 1,867 meters. And for the small hills and plains, they are occupied about 60% of the whole Hainan Island. Generally, we know that is on the list here is the south part of Hainan Island. Tomorrow we will go there. And for the climate, the Hainan Island, the typical climate is tropical monsoon climate. There are also dry and rainy seasons. May to October is the rainy season, and November to July, November to April next year is the dry season. The annual rainfall varies from 1,500 and 2,500 millimeters. The annual temperature is about 23 to 25 degrees. And Hainan is one island is really affected by typhoon in summer and autumn. And the annual typhoons to the land in to the island, influence island is about 3 times per year. And there are several different vegetation types in Hainan Island. From here, this is the vertical distribution of the vegetation types in Hainan Island. This is, I list the Jianfenlin's vegetation as an example. The highest part is the mountain evergreen broadleaf forest, and then the second, the tropical mountain rainforest. And then below is we call the lowland rainforest. Is composed of 2 vegetation types. One is the tropical semi-deciduous monsoon forest. Another one is tropical evergreen monsoon forest. And the fifth one is the savanna and shrub. The elevation is very low. The typical Diptocapsule, typical arches species distributed in the lowland forest. But the tropical mountain rainforest has the largest forest areas. And this is the spatial distribution of the nature forest in Hainan Island. Professor Liu also introduced this morning. The forest coverage of the whole Hainan is about 62. And these are the typical landscapes in Hainan Island. On the left part and top of this is Jianfenlin. There are many big trees and ferns and lianas and palms in Hainan Island. And Hainan Island is also one of the most hotspot area of the richest biodiversity around the world. We can find here. This is the Shannon diversity. We can find the Hainan Island the diversity is very close to Brazil and higher than Burma. And this is the species composition in Hainan Island. We can find that for the plants, there are more than 5,400 species in Hainan Island. There are also many different species here. And this morning, Professor also already showed the list that the Hainan Island from 1950s to 1993, natural forest area decreased rapidly. But since the 1993, the government of Hainan province decided to start logging the tropical forest, and the second forest area is increasing the outlet. And we also built a state key field research station in the Jianfenlin. And in the Jianfenlin list, our institute was located in Jianfenlin. We started field investigation from 19 1957. And so this is the 10th, and the day after tomorrow we will visit the lab. We cooperate with the local government, the Jianfeng National Forest Reserve and National Forest Park to monitor the forest. This is the 10th, it's man-made, it's not natural lake. And this morning, Professor Liu also already told the list that we established the series of plots in Jianfeng. We have two big plots. This is the two biggest plots in the world. One is the 60 hectare plot. It's established in 2012. And the another one is 64 hectare. It's in the secondary. It's established 5 years ago. So this 6 hectare is in the primary. Another one is in the secondary. We also have many small satellite plots that are surrounding these two big plots. We measure the soil, we measure the microorganisms, we sample functional traits for all species across all of these plots, especially for the small satellite plot. We sample the leaves for each species in each quadrant. And this is the topography of our 60 hectare plot. And this is the soil's available nitrogen distribution in our 6 hectare plot. You can find that from the left part, the soil's nitrogen is relatively low, but to the right top is part, the soil nitrogen increase is high here. And this is the interesting pattern for the soil nitrogen. We can also find that for the legumes, because legumes can fix the nitrogen, and the red circles are the Amosia. Amosia is one genus of legumes. And the black and the blue circles are the Archidendral. This is another legumes genus in Fabaceae. But these two legumes groups, species groups, they utilize the different soil nitrogen habitats. Amosia prefer the low nitrogen area, but Archidendral, this genus, they prefer the high soil nitrogen habitats. So, and we also, the Jianfeng 6 hectare plot is also a part of the Forest GEO's network. We collaborate and publish several papers. And in 2016, the Forest GEO's data analysis workshop was held in Jianfeng. There are about 70 scientists went to our plot and worked there for 2 weeks. And Steel Harbor and Fanlianghe, they also visited our plot for several times. And in our plot, we monitor the typhoon impacts on the forest. I just mentioned that there are about 3 times the typhoon past the Hainan Island. The typhoon also has a great influence on our plot. This is the plot that was influenced by typhoon about in 2013, about 12 years ago. So we are still monitor the typhoon's impacts. Sometimes there are strong typhoon. After that, we don't go into the forest to measure the trees that were to learn how many trees were blown down by the typhoon. And this is our study based on the satellite grid plots. We study how the secondary forest recovered half centuries after logging. We found that the species region is recovered quickly, but not the basal area and the biomass and the forest structure. They need more time to recover. And we also study how the red species on common species distribute along the soils nitrogen and phosphorus gradients. We found that the red species preferred preferred high phosphorus habitats, but the common species preferred the high nitrogen habitats. And common species and rare species also show the negative relationships. And for the legumes, because you just find that you can see that they are very clear the distribution pattern for the legumes. So we also study the soil to study how the soil's nitrogen concentration can mediate the relationship between legumes and the neighbor diversities. We found that where soil's nitrogen is high, most legume species have a higher neighbor diversity than non-legumes. And where soil nitrogen is low, most legumes have a lower neighbor diversity than legumes. This is based on the 12 big plots across the world. And in Jemberlin, we also studied can the old growth tropical forest sequester the CO2. We found that the Jemberlin's old growth forest is the net carbon sink. Even though there are many primary forests in Jemberlin, but most of the forests in Jemberlin are secondary. And because our is the for Jemberlin is the ecological monitoring station, so we also monitor the climate changes. This is the climate changes from 1980 to 2015. We found that especially for the average temperature and average land temperature, they increased gradually in the past several decades. And the warming index also increased. And for the second part, I will just share our one new findings about animal behavior. It's a new behavior. And we know that the squirrels can disperse the nuts. They can store the nuts on the trees. And storing food is a common species to specific behavior used by the squirrels. But for most squirrels in the temperate forest, the squirrels, they just hoard the nuts under the leaf litter in the hole in trees or logs in the ground. But in subtropical or the tropical zones, nuts hanging on the tree's branch behavior thought to maybe can minimize the fungal infection in humid environment, especially in the tropical forest, or decrease the risk of loss through decomposition or germination. In Jemberlin, mid tomorrow, the day after tomorrow, you can see these kinds of nuts. They were hanging on the trees between the twigs. You can find that this is the two different Fagaceae species nuts. One Cyclonopsis nuts. One is the oval. Another there are two different kinds of nuts types. And we found that these kind of special nuts were stored on different plants. For most are on the alive trees, and some are on the dead trees, and on the alive liana and dead liana, and few on the bamboo. So for this special nuts farmers, we have one question. Because we know that there are 9 squirrels in Hainan Island, so we want to ask whether some of the 9 squirrels identified from Hainan, the tropical forest, use the special behaviors to prepare these special nuts and fix them securely on the vegetation. And after the detailed field investigation, we found that the 2 Fagaceae species is the Cyclonopsis plants and 2 squirrels storing the nuts. So these 2 study sites are our 2 big plots. One is 6 hectares and another is 64 hectares. And we put many camera in the field, so we know which kind of the squirrels they store the latent night. This is two kinds of the squirrels. These two kinds of squirrels that we identified by the camera. We found that they are smaller in size and arboreal, and they are good at climbing and able to create many feet on the seed of acacia. And this is their body length. Their body length is not the largest one and also not the smallest one. And this is the one squirrel. This squirrel is mainly distributed in Myanmar, Thailand, Laos, and Vietnam. And another squirrel is mainly distributed from Vietnam, Laos, and Cambodia with the southern side of China. And this is the two Cyclonopsis species that the squirrels stored on the twigs. One is the Cyclonopsis edicea, and the other one is the Cyclonopsis platyliformis. And these two Cyclonopsis species are also the most abundant species in our big plot. In the big plot, you can find the Cyclonopsis edicea has the 1,600 individuals in our plot. For our plot, you previously saw this morning, we have about half a million individuals in our plot. So these two species are the most abundantly used and stored by the squirrels. And this is the Cyclonopsis. You can find that there are some, the squirrels cover the middle of the nuts and then put them just between the twigs. It's a really special. But for this is oval nuts, but for the other kinds of the nuts, you can find that they are severally covered. Here, they are asymmetric, there are two. So they just cover the nuts and then place the nuts between the small twigs. And the position of these two nuts being covered and fixed are different. And we can find that for this oval, in the middle, there is one groove. Sometimes there is only one groove. And sometimes there may be two or spiral groove, and sometimes there are two separate grooves for this kind of nuts. But for the other different kinds of nuts, this one, they may cover at the bottom. They cover at the bottom, not in the middle. So at the bottom of the nuts, they are 10 symmetric or random distribute the grooves. And the squirrels, the grooves on the surface of the fruit, all the squirrels to firmly fix the fruits of these two species of Cyclonopsis. In a way similar to the Maltese and Tendon-Jones. This is a typical architecture in China. The squirrel used these two twigs as a raised tendon-like structure to fix the groove Maltese-like structure covered on the surface of the nuts. So the squirrels can cover the grooves on the fruit based on the various forms according to the fruit shape. And we also study the number of grooves, the distribution, these two figures, so the distribution of two figures. And we found that for these two species, they have a different the number of grooves. For the oval-shaped nuts with two unconnected or spiral-shaped grooves seem to help adjust the storage position of the nuts to fit the spatial direction of the branch. But for the another nut, a symmetric bottom groove may help squirrels to more easily secure nuts when rotating, optimizing the nut fixation position for the safety fixation between tree branch. And this is we also study the width of the grooves. And we found that the grooves width of the nuts match the typical width of the squirrel para-incisors. And we found that we observed the effect made by squirrel in testing and adjusting nuts to be more tightly fixed, which suggests that the squirrels, they may have a proactive cognitive process in this storage behavior. And these nuts can be stored on the twigs really firmly. You can find that we shake the branches, then the nuts cannot fall. And the trees stored the nuts will also have different characters. And the tree has where the nuts are stored mainly between the 1 to 2.5 meter. And the DBH for trees to store the nuts mostly on small trees with the DBH rate from 0.4 to 1.6 centimeter. And we also found that the nuts were stored between a branch with diameter between 0.1 to 0.6 centimeter. And nuts are mainly fixed between two small branches with a branch angle of 25 to 40 degrees. And I will also show you some behavior videos for storing nuts. This is the videos to show how the squirrels to removing and placing the nuts. You can find the little squirrel here. This is the nuts. They were placing the nuts. And then the nuts were then removed by the squirrels. I will also show you videos to see how the squirrels cover the nuts with the teeth. This is the squirrels. They were covering the nuts. So they will try it several times to fix the nuts between the twigs. And this is the process of the using teeth to reinforce the storage of the nuts. You can see that these two types of squirrels are using their teeth to reinforce the fruit stored on the branch so that the nuts can be more stably fixed between the branches. And the last video is about how the process of removing the nuts. During a period of more than 4.5 months, we captured 47 images from 32 infrared cameras. And this is the nut storage behavior is very common in Hainan Island. And this storage behavior also has impacts on the plant diversity. We can find that this is the nuts being covered, but sometimes maybe after long times the squirrel didn't go back to take the nuts. The nuts fall on the ground and then the seeds will germinate. And the store nuts may be also destroyed by the insects. You can find that there are small holes on the nuts, so some insects bite the nuts. We also studied where the nuts were stored on the trees. We found that the nuts were mostly stored on the trees with distance to the parent trees. There is about 10 to 25 meters. You can find the nuts were not stored near the parent tree, but they are stored far away from the parent trees. We also study how the number of seedlings of these two species germinate. We found that the number of seedlings increased significantly at distance 15 to 20 meters to the parent trees. So this figure is corresponding to the previous figure because there are more seeds were moved from the parent trees to far away from the parent trees. So there are more chances for the nuts to be fall down and then germinate. So there is the hamper here. The black one is the below the squirrel activities. The red one has the squirrel activities. So for the parent trees with the squirrel activities, when the squirrels they disperse the nuts, there are more possibilities for nuts to be germinated far away from the parent trees. And but for the next study, some scientists they all wrote some comments on our paper. They may want to know how this special animal behavior will learn from the parents to the next generation, or how they learn each other in between the different squirrels. But I'm not I know plants, but not animals. So maybe it's welcome for other scientists to study this special behavior in Hainan Island. Yeah. And that's all. Thank you.
Very interesting talk. And when I was young, I was taught that the difference between human beings and animals, we know how to make tools and use the tools. Apparently, I think the knowledge was only partial. Okay, right. And we have time for a few questions. Dr. Han is not only ecologist, he's sound, and he's botanist. Now he's becoming animal scientist. He's going to become, okay, Richard?
I would imagine they steal each other's nuts. I mean, are they territorial? Do they defend their nuts?
Defend
Defend their nuts from other squirrels?
defend the nuts.
Because there are a lot of birds store acorns and they steal each other's acorns, for example.
For example,
And
so,
jays do that.
yeah,
And I would expect that they're territorial and a squirrel is defending an area to prevent other squirrels from stealing its nuts.
yeah. Sometimes they may defend for this tree. Do you mean this tree? This one? They will store the nuts just far away from parent trees. So they defend. I think it's the defender for other animals to be these nuts to be stolen by other animals. Yeah. And for the nuts because I just saw that the nuts were stored on the trees at 1 to 2.5 meters. They will also defend the other such as the mouse and small animals to steal these nuts.
I think to answer the question is, do you have data that the number of nuts stored by a squirrel, then the number of nuts retrieved by the squirrel, do you have such data?
Yeah, I have this kind of data.
Like equal? The low sum?
Lost. How you mean how many proportion of the
say
nuts were lost?
they put out 100 end of the year, they only retrieved about 50 because other half have been stolen or.
And actually, I deleted that page, but I remember that they are about 30% of
loss.
Lost, but maybe lost. Maybe there are many causes. Maybe being stolen by animals or just fallen because of their also very strong typhoon there. Maybe we do not know. Many different causes. Yeah.
Yeah. Any other question? Because interesting also the story I heard from northern China, and the squirrel had very bad memory. They buried nuts various places, and then they forgot where they buried them all. So that's where the Korean pine regenerate because from escape to seed. And that's a story I heard. And you did not have experience. Yes, Ben.
Technical question. Soil data. A technical question about your soil data, because you showed some nitrogen data for the plot, but you didn't have a unit, but I assume it's milligrams per kilogram. It seemed very high. So I wondered what that data was. You said available nitrogen, but
or available nitrogen. That's available nitrogen distribution map.
yeah. So what was the method for measuring available nitrogen? Because those concentrations are extremely high for a tropical forest.
我们的叫
If it's what I think, I mean,
it's measured by the traditional level method.
KCl extraction or something.
Yes, it's KCl maybe.
Okay.
Yeah.
Yeah. So they seem extremely high. So usually KCl nitrate or ammonium would be 1 or 2 milligrams per kilogram, and you had like 300 or something. So I wonder if there's some kind of storage effects like contributing to the high nitrogen.
Yeah.
Like, so if you store the soils for a long time before you extract them, you really increase the nitrogen availability or the apparent. So I just wondered what. We can talk about it later.
Yeah,
But
yeah.
it's KCl extractable nitrogen.
Yeah, we can talk about that if you want to know
interested.
more about our soil's nitrogen
Thank you.
levels.
Also for the site, it's very diverse. Have lots of species and lots of nitrogen fixing species, but I'm not sure that's have anything to do with high nitrogen level. Yeah. But we find out tomorrow when we get down to the Jianfenli.
You will go to Germany. I can show you some legume species. You can find some species prefer low nitrogen and some prefer the high.
Okay. Question from the back? No? Okay.
Okay.
Thanks for save us some time. And so,
Thank you.
okay, thanks. You. And so, last but not least speaker, and this Dr. Jianxin from School of Forestry from our university, Beijing Forestry University. And he's recently joined our university. But before coming here, he has wide experience. He got his PhD from Institute of Remote Sensing and Digital Earth of Chinese Academy of Science. And since then, he been working as postdoc at 2 of the top universities in China, Beijing University and Tsinghua. That's some place we dreamed of going. And he also had experience, research experience in UK, Imperial College, London, and also in Netherlands. Okay, now I'll hand over to Dr. Tan.
Thank you. I have to apologize because I don't have any interesting videos with squirrels. My name is Jianxin, and I come from the College of Forestry and Beijing Forestry University. Because I am the last one speaker of today, so I want to thank you guys for being here, and thank you for your patience and suggestions for the presentations. So before the presentation, I would like to introduce our team. My presentation will be about modeling of forest carbon and water flux based on the eco-evolutionary optimality theory. So the prototype of this modeling framework was established by Colin from Imperial College London and Sandy from Reading. And I collaborate with Wang Han and Zhang Yao from Tsinghua and Peking University respectively during my postdoc period. And now I'm working in Beijing Forestry University and collaborating with Huang Huaguo because our team has a background of remote sensing simulation based on 3-dimensional radiative transfer model. So my current work is planning to combine the state-of-the-art remote sensing simulation method and traditional ecology theory to improve the estimation of ecosystem carbon and water flux. During the modeling, we also have the contribution from the Brown University and UC Berkeley. So first of the presentation is the background. As we know that we always use gross primary productivity to represent the ecosystem photosynthesis rate, And because all plants and all ecosystems are alive because of the photosynthesis process, so we usually think that the GPP forms the primary terrestrial carbon uptake flux. And because we hope to predict the plant dynamic in the future and what the climate will change in the future. So predicting the gross primary productivity, the GPP, is very important in predicting the plant growth and the plant dynamic in the future. And the evapotranspiration is a combined word that contains the evaporation from soil and from the intercepted water and the transpiration from the vegetation. And the evapotranspiration forms the primary terrestrial water consumption, that all of the terrestrial water from liquid condition converted into the water vapor and runs into the atmosphere through the evapotranspiration. About 70% of the precipitation on the terrain transferred into the go back to the atmosphere by the evapotranspiration process. So if we hope to better predict the climate change in the future and the plant dynamic, plant growth in the future, or forest growth in the future, we have to improve the GPP estimation. And the evapotranspiration helps us to understand the water constraint from the environment on the plant growth. So GPP and ET is very important for ecology and for geography studies. But unfortunately, there are still negligible uncertainties within the GPP models and within the evapotranspiration models. I give you some examples here. The first example is from a paper on GMD in 2020. And Benjamin Stoker wants to compare the uncertainties from different GPP models, and you can see here there are huge uncertainties between different GPP products, or based on remote sensing or based on land surface models. Part of this uncertainty comes from the different data, the different input data sets from the to describe the meteorological condition, but there are still huge uncertainties within different GPP models. But when we hope to predict the GPP trend in the future, the uncertainty increases, because here is a study from 2014. And we can see here, if we want to predict the GPP trend, the photosynthesis trend in the future, some of the models predicted there is an increased trend of the GPP, but some of the models predicted there will be a decreased trend of the GPP. So we can see there is significant uncertainties within current GPP models. Also, here is a very, very recent study published on Nature Communication in 2024. Our colleagues employed observation from 3 tropical forest sites in Ghana, in the Africa, and they find that because the tropical forest, because we have the workshop for tropical forest today. Tropical forest is the most productive ecosystem in the world because it has a very sufficient water supplement and radiation. But there is a very, very common underestimation of the gross primary productivity within these 3 tropical forest sites. And we can see here about 20% to maybe 50% of the underestimation has been observed in these 3 tropical forest sites. So we are wondering if we can improve the model. And the uncertainty is also observed in all current ET models. For the current estimation, here is a paper published on Nature Reviews recently. There is uncertainties between current ET products to estimate you can see here 1980s to 2020, different models exhibit same increasing trend but different magnitude of the evapotranspiration on Earth. And if we want to predict the future, the evapotranspiration also exhibits an increasing trend. But you can see here the standard error. There is uncertainty within different ET products and ET models. And when we try to explain these uncertainties and we try to deal with the uncertainties, we find that two major reasons contribute to this to current models uncertainties. The first reason is the parameterization modeling. During the estimation and the modeling tasks, most of the modelers try to divide the continuous varying terrain surface into discrete types, such as the evergreen needle-leaf forests, evergreen broadleaf forests, the plant functional types, and in some studies the land cover types, and give different types some different parameters to describe the traits of this type of ecosystem, but we cannot expect a 100% accuracy of the land cover project. Usually, the accuracy of land cover products is about 80%. So we can see here, if we use the same input data into a model, inputting a model and calculate the canopy conductance, and we use the different parameterization strategy for different land cover or plant functional types, we will receive very different result of the conductance estimation. This is just an example from my own study. And there is another evidence, because we are facing this changing climate, the air temperature is increasing and the CO2 concentration is also increasing. But if we use the varying temperature and the CO2 concentration in the model, different models exhibit diverse sensitivity to the changing environmental factors. So this uncertainty is mainly caused by the parameterization modeling and the model structure itself, because different models exhibit different sensitivities. So this is the second reason, because I have a background of remote sensing, so I am more interested in the radiative transfer process in the canopy. So this study, this recent study suggests that except for trace estimation, the VCMax estimation error during the modeling and the neglection of the optimality theory that I will talking about later. Here is a significant uncertainty comes from the simplification of the canopy radiation, because most of the current GPP and the evapotranspiration modeling are using the remote sensing observation of the canopy, remote sensing observation directly, but not simulate the radiative transfer process detailedly. So you can see here a significant part of uncertainty comes from this simplification. So this is why what we want to do, we hope to include this process and describe this process into the GPP and the ET modeling to improve the estimation results. So the second part of the presentation is the theory basis of the eco-evolutionary optimality theory. Because we have the audience has a different background, so I try to introduce this part more briefly and in plain language. So what we have done is what we have done based on two hypotheses. The first hypothesis is the least cost theory, which was first proposed by Cohen and Farquhar in 1977. Because we know that if the plant want to do the photosynthesis process, they must open the stomata to exchange the CO2 and the water with the atmosphere. So this theory is talking about the plants tends to maximize the marginal carbon gain while minimize the marginal water loss, because they want to save water. They don't want to lost more water than expected because most of the plants are suffering with water stress. So we can then come up with this equation that the plants tend to achieve the marginal water cost. And then we can translate this all sentences into this equation. And this is the first hypothesis, the least cost theory, because the plant want a least cost of the water. And this process is achieved by the leaf stomata behavior, and the leaf can acclimate to the meteorological condition by itself to achieve this optimality criterion. And the second hypothesis is the coordination theory. We know that the apparent photosynthesis process is determined by the two reactions. That is the light reaction and the carboxylation. Because this theory has been proposed by Farquhar. And the second theory is that on a long period, the plants tend to save the energy and do not want to waste the energy to maintain the carbon oxidation maintenance. That is, on a long period, the plants tend to acclimate to the environmental condition and let the carboxylation rate equal to the light reaction, and we can then write this equation that apparent photosynthesis rate is equal to the carboxylation rate and equal to the light reaction rate, because the carboxylation capacity acclimate to the prevailing radiation, and so that it can acclimate to the recent environmental condition. And this theory was first proposed by Collin in 2014, and then evaluated by Wang Han in 2007. And then, if we reconcile these two hypotheses and some other equations, we then come up with this P model. The P model is a universal model for all C3 plants. That means we do not need to calibrate the parameters for different plant functional types or different land cover types anymore to give them specific values as the parameters. The model is universal for all C3 plants, and we did some simplification to extend this model to enable the model can work for the C4 plants. And the model has a formulation of the light has a structure of the light use efficiency model. That means the blue part of this model represents the light use efficiency, and the red part of the model describe the energy, the radiation energy absorbed by the canopy. Because here, if we want to estimate the photosynthesis, we can estimate the canopy absorbed radiation and the light use efficiency independently. This model structure has some advantages. Firstly, because if we want to estimate the GPP for regional, if we want to estimate the regional GPP or global GPP, we hope to employ the remote sensing data sets, and the light use efficiency is very easy to combine with remote sensing inputs. And secondly, because we have two independent parts of this model, so we can calibrate, we can evaluate the two branches branches of the model independently to improve this model. This is what I have done in the following slides. So to improve the estimation of the global photosynthesis and global evapotranspiration. This model was first proposed by Wang Han on Nature Plants, and then evaluated by Benjamin Stoker from the Brown University. So this is the first part of the research. We employ the P model and the model extension to estimate global photosynthesis and global evapotranspiration. Okay, first, this is the P model equation. It is universal for C3 plants and for C4 plants. And here is a global evaluation of the only one parameter. This parameter is not is the same for all C3 plants. They just need one parameter to describe the intrinsic water use efficiency. And Wang Han has evaluated this parameter by a global independent data set based on isotope observation. So the users do not need to calibrate or evaluate calibrate the parameters in these models anymore. And this is my work. I extended this model to estimate evapotranspiration and the water consumption, based on the remote sensing input. Firstly, because current remote sensing-based evapotranspiration models mostly use the Peyman-Montes equation, and they need the estimation of canopy conductance to describe how the leaf stomata constrain the water diffusion from the leaf internal to the atmosphere. And existing models also utilizes always utilizes empirical model or parameterization model, includes some parameters for different types. And I am using a physical-based model, the fixed law. The fixed law is a physical model to describe the diffusion capacity, the diffusion efficiency of different molecules. That is the water molecule and the carbon dioxide between the leaf internal and the atmosphere. And then the key factor of the fixed law, we need the ratio of leaf internal carbon dioxide intensity and the carbon dioxide intensity of the atmosphere. We need this ratio, and this ratio can be estimated based on P model. And this ratio is the driving force of the CO2 and the water diffusion of the water diffusion process. And then we can estimate the transpiration rate based on the Peyman-Montes equation, and then we can calculate the evapotranspiration by including the soil evaporation, including the soil evaporation, because the soil evaporation is a very complex process, and it determines by a lot of things, by radiation, by the soil texture, by the soil moisture. So we need, we here employed a ratio for each site that estimated based on environmental conditions to reduce the parameter amount of this model. And here is the model, is the workflow of this model. This part describes the equations here, and I want to see. What I want to say is that because I want to estimate evapotranspiration global-wide and the GPP global-wide, so I have to do a lot of calculation task. So I employed the Google's engine platform to finish the computation tasks, and we finally can generate a 500 meters grade GPP and ET products with 8-day time step covering the entire world. And then we can do the the evaluation task based on the FluxNet sites. We import over 100 global distributed FluxNet sites to then the site scale evaluation, and then we can do the basin scale evaluation to test if the model has a reasonable performance in spatial extrapolation, and then we can detect the temporal trend of evapotranspiration and the gross primary productivity. And here is the result of the GPP. So the first part is the site scale evaluation. We compared our predicted GPP against the FluxNet observation, and our model achieves very satisfactory site scale evaluation results. And this result is the same, is nearest the performance of other GPP products, and we have no type-specific parameters to be calibrated. So it is more convenient for the users to employ our model in GPP estimation than use existing other parameterization models. And here is a comparison of global pattern of the evapotranspiration. The first one is our result, and the second one is from MODIS and from PML. And the third one, the final one is from the SIF observation. The SIF is the shot of the solar-induced fluorescence, and this special remote sensing observation represents a direct observation of the photosynthesis rate. So here we can see there is a very similar global trend between our estimation and other GPP products and the SIF observation. This means our model can achieve a very satisfactory reasonable global pattern against other products. And here is another interesting result. You can see here clearly. When we hope to predict the future trend of the gross primary productivity, we received a nearly same increasing slope of the GPP against the ensembled result of the Trendy. Trendy is a ensembled data set that contains several vegetation growth models to predict the future dynamics. So this result means that our model can achieve a very reasonable result against the climate change. So we can see our model is a reasonable GPP estimation model. And here is some of the ET results. The first part is also the site scale evaluation. We compared the evapotranspiration estimation against the site scale flux observation. And the second part is the global pattern comparison. We compared our estimation against most of the evapotranspiration products from based on remote sensing and based on land surface models. And we received a comparable trend of the temporal trend of the evapotranspiration against the model average between all other models. And finally, here is a very interesting result, because we don't have the type-specific parameters, and this means our model can be extrapolated to the spatially extrapolated without any calibration work, and we theoretically we can receive a better result during the spatial extrapolation. And here we employed a basin scale water budget closure method to evaluate our method. The evapotranspiration is calculated based on the basin scale water budget, the precipitation minus the runoff minus the discharge of the groundwater. And here is a very interesting result that our model received satisfactory performance against the water balance. This means this performance is the state-of-the-art performance against existing evapotranspiration products. This means our model has a reasonable performance in predicting the evapotranspiration in non-size calibrated regions and in the future. And our model has also had some applications after the publishment. The first part, because we can estimate the photosynthesis rate with a reasonable performance. So this means we can predict the photosynthesis related traits based on this theory. So this application is mainly about the estimation, the prediction of ecosystem carbon oxidation capacity. That is the VC-max, and this trait can be predicted based on the P model. And here is the result published on Ecology Letters from Nicholas Smith, and they received a satisfactory performance in predicting the carbon oxidation capacity. And this result is comparable against the field investigation. And a recent study, I can't remember the published date, suggests that our model can generate a comparable VC-max prediction against a very, very complex remote sensing based product. That means the theory is reasonable in photosynthesis traits prediction. And a very recent study in this year, our colleague demonstrated that the land surface models can be benefited from the VC-max prediction because we can predict the carbon oxidation capacity, and then we can employ this capacity, this trait into the land surface models to predict plant growth and to predict the land surface processes. And so we can see the land surface models can be benefited from this prediction. And secondly, because most of the plant growth models, including the crop models, the tree growth models, and the dynamic global vegetation models, were built based on the photosynthesis, because the photosynthesis provide the material basis for plant growth. So we can replace the GPP estimation for plant growth for current plant growth models, and we can then incorporate the water limitation based on the evapotranspiration estimation in the growth prediction to predict the plant growth for cropland and for natural vegetation. And here we received a satisfactory performance in predicting the wheat yield in China because the cropland is managed by human beings. So cropland is different with the natural vegetation. And here is an example of natural vegetation growth prediction. We did this experiment in the Tibet region of China, and we can predict the peak growth of vegetation in this region, and we have a good correlation with the remote sensing observation. And this tool has been published several years ago. And good news is that this optimality theory can be further applied into the plant trees prediction. Very recent study has been accepted by science. And to predict the stem photosynthesis rate and stem respiration based on this theory. So I mean, the eco-evolutionary optimality theory is a very useful theory to support the plant trees prediction. And the second part is the plant optimality behavior. During the first part, when we are talking about plant optimality theory, we actually do not know how the plant acclimates to recent environmental conditions. When we are estimating the GPP and the evapotranspiration, we just use the average climate condition of the past 8 days or the past 12 days or the past 15 days. We just use a direct averaging environmental condition. But we know that the photosynthesis and evapotranspiration is an enzyme-driven reaction, and the enzyme requires a period to acclimate to recent climate change, to recent environmental condition. That means the environmental condition should not perform an equal effect on the predicting of the light use efficiency. Here, light use efficiency is an example. There should be a weighting curve of the climate condition within different time step. This is an example from the past 30 days. The weighting curve multiplied by the environmental condition in the past 30 days, and then we can receive the light use efficiency that acclimates to recent environmental condition. So this should be noted that the antecedent condition has a legacy effect on the light use efficiency. And Kiona from the Arizona University has reported this phenomenon, and they wrote this as the ecosystem memory. But their studies employ the GPP and the evapotranspiration as the target, and they do not detect the optimality behavior of the light use efficiency independently. So up to now, there is no study reported how to describe this optimality behavior, and if we include this optimality behavior, can really improve the GPP and the evapotranspiration modeling. And here is their study that they reported that here is a considerable part of the accuracy can be improved by including the ecosystem memory into the consideration. And here is what I have done recently. I hope to decompose the different weights from different time steps to figure out a clear optimality behavior of different ecosystem, and I want to check if this optimality behavior can be substantially support the GPP and GPP modeling in this study. So I employed a method of interpretive deep learning network to finish this study. So the first time is that the first step is that I employ the temperature, the vapor pressure deficit, and the soil moisture to predict the light use efficiency, because we think the light use efficiency is mainly determined by these three environmental factors, and I cut all the entire time series environmental factors into the slice with 30 days, and then we train the LSTM, the deep learning network. LSTM is the long short-term memory deep learning network, and this network works for time series data prediction, and we train this network to predict the light use efficiency here, and we received a very satisfactory light use efficiency prediction. And finally, we use an interpretive algorithm, the expectation gradient algorithm, to decompose this trained network, and we can decompose the contribution of different time steps, and then finally we can reach this weight curve of different for different input factors, and then we believe that this weight curve represents the optimality behavior of different ecosystems. Here is the results. It is very interesting results because we observed two typical optimality regimes. The first part is very reasonable. We can easily to understand that some of the ecosystems are more sensitive to the climate condition in the recent 5 days. You can see here the weights from recent 5 days takes up about more than 60% of the overall weights to the final light use efficiency prediction. And some of the sites belong to the regime 2. And we can see there is no significant difference between the between the optimality weights of the past 30 days. And we can see the ecosystem from the regime 1 has a stronger optimality capacity to recent climate environmental condition, and the ecosystem belonging to the regime 2 has a weaker optimality capacity to recent climate conditions. And we also find a very interesting phenomenon that most of the regime 1 ecosystem locates in the wet environment. The precipitation and the aridity index significantly surpass the ecosystems in the regime 2. So this is a very interesting phenomenon. And we can predict whether ecosystem belongs to the regime 1 or regime 2 based on the environmental conditions. And I'm not showing here, but we can predict if the ecosystem belongs to each of one. And then because this prediction should be done because we want to predict the gross primary productivity over the world. So we need to do this prediction. And then we incorporate this optimality behavior, the predicted curve and the predicted regime into the GPP modeling, and we received a very common improvement of the R-square and the relative RMSE improvement of the GPP prediction. So without any other input, information, just incorporate the just replace the climate condition by the optimality, by the convolution result of the weight between the weight and the recent climate condition. We can receive a significant increase of the resulting accuracy. So this is a very interesting study to demonstrate how the different ecosystems acclimates to recent climate conditions, and is there any difference between the optimality behavior? And here is another interesting thing that most of the current deep learning studies in ecology or in botany like to describe the deep learning algorithm as a black box, and someone do not like this black box because it provides no insights into the process. And some recent studies demonstrate that we can guide the deep learning models by incorporating some knowledge-informed neural network, knowledge-guided neural network. And our study is another strategy of applying the deep learning network in ecology studies. We use the interpretive deep learning algorithms to show some to provide some information of the to support our ecology modeling tasks. And here is the final part of our study. Is a simulation of the canopy radiation transfer process. The data comes from our field investigation. So we scanned entire forest ecosystem based on the terrestrial LiDAR system. Some of our colleagues have introduced that we have some observations from the airborne LiDAR system, but what we are employing is the terrestrial LiDAR system because it can provide a smaller but more fine-grained three-dimensional observation of the forest ecosystem. And then we can rebuild the three-dimensional forest scenario based on state-of-the-art radiative transfer model. The three-dimensional transfer model can then, here is the result of the scenario building results. And then we can simulate the canopy, the radiation absorbed by the canopy, and this radiation then participate in the photosynthesis process. And the incident radiation, the incidence radius angle can be set according to the time step because the solar zenith and the solar position and the incidence radiation angle is different according to the local time, and we can set it in the software. And then we can simulate the radiation transfer process for the entire ecosystem and for each single tree, and even for each leaves. Even though the leaf is not the real leaf in the forest system, but we can simulate the radiation absorbed by each single tree based on this radiation transfer model. And finally, the energy absorbed by each tree and the canopy can be estimated to support the downstream GBP modeling and evapotranspiration modeling. And you can see this flash because we can set the incidence angle of different time step here. Here is the time step, and then the energy absorbed by each single tree can be then simulated by this model, and we can estimate the hourly energy absorbed and even minutely. But this is not useful in modeling framework. So we just use the hourly energy absorbed by each single tree to support the modeling. And we observed a huge bias between the simulation results against the satellite remote sensing observation. And the error transmitted determined by the weather condition can also be configured during the simulation process. And what we are planning to do is to extend our method to scan the canopy structure for some for some flux sets, especially the flux sets in tropical forest regions, because the tropical forest region has a very complex multi-layer multiple canopy layers. So we hope to collect data from these flux sites and then simulate the daily or even hourly radiation and gross primary productivity and evapotranspiration for these sites to see if our model really works to support the flux modeling. So that's all. Thank you.
Okay, thanks. And because of time, we will not have a question for this talk, but if people are interested, can contact him and talk after the conference. And let's give a big applause to all the speakers today and for the diligent work and informative talk. Okay. And that concludes our scientific presentation for today. But before we end the conference, this workshop, we have a special session, final special session, unless the editorial board have another session. But I would like to invite joint editor-in-chief and my colleague, my friend, and John Kershaw, and to have a special presentation.
Thank you very much. And I would like to, at this point, ask Huang and Wei Li to come up. These two colleagues are the powerhouse behind the journal, behind the workshop. They keep Jianxin and I out of trouble, and many of you out of trouble by tracking where the manuscripts are. The logistics behind this is phenomenal. Their help is phenomenal, and they're really two of our best friends, I think. And we want to take just a few minutes to thank them. So maybe a round of applause for them. And then I also have a little gifts that I brought from Canada for each of them. So for you, and this one is for you. Thank you. So thank you very much, and that concludes the workshop, and hope to see everyone again next year. So
Next year.
Okay, thanks, John. And John brought the gifts all the way from Canada. So I'd like to thank John for this nice gesture on behalf of our editorial board. My job is done.
Yeah, okay, thank you. Thank you all of you to come here to support us. Today's presentation is the end. So tomorrow our key speakers and our editorial board will go for another field trip. 非常感谢来的各位老师和同学，然后希望明年的时候大家继续支持我们，感谢大家。